there we go. So this is the paper we're going to talk about today. I did put the PDF as a pin in the chat so you can read it for free. You don't need any sort of account. So basically I'm going to kind of the way I'm going to go through this paper is I'll read certain blocks of text, typically the ones that I've highlighted because those are the important bits of text. Then we're going to get into the figures. Now I think one of the most scary things, at least for me, when I first started reading scientific papers was trying to understand what the hell they're even talking about here in these figures. You're probably looking at this and I think most of you can recognize that this is a mouse, but everything else looks like gibberish. And I've made some notes for myself when I read this paper to kind of help jog my memory, because as I read a paper, I sometimes forget what the point of a figure was. But we'll go through this data together and hopefully it's not too scary. We might not even get through the paper because I do want to explain lots of things as we go through it. Because I take for granted a lot of the words that are used in this paper, but I realize most people might not understand what I'm saying. The title of this paper is Enterococcus, get your minds out of the gutter, peptidoglycan remodeling promotes checkpoint inhibitor cancer immunotherapy. So what is Enterococcus? Enterococcus are a type of bacterial species that normally live in your gut microbiome. There's a bunch of different types like E. facialis and E. faecium. And let me just see if I'll just put the Wikipedia link for this into chat so you can look up Enterococcus on your own. Normally they're pretty harmless bacteria. They're just there doing their thing in your gut, being helpful. But sometimes they can cause disease. So I'm reading something on Healthline, which is just the first article I pulled up, and I'll put the link here so you can read along with me. I'm not showing it on the screen. Basically, Enterococcus faecalis, which is actually one of the species that are going to be talked about in this paper, is a species of bacteria that is naturally found in our intestines. While it tends to be harmless in that environment, it has the potential to cause a serious infection if it spreads to other areas of the body. Enterococci, which is the plural form for the species, are a type of bacteria that live in your GI tract. There are at least 18 different species of these bacteria. E. faecalis is one of the most common species. These bacteria also live in the mouth and the vagina. They're very resilient, so they can survive hot, salty, or acidic environments. I'm sorry, I'm keeping a straight face. E. faecalis normally live harmlessly in your intestines. However, if it spreads to other parts of your body, it can cause a more serious infection if the bacteria get into your blood, urine, or a wound during surgery. From there, it can spread to different sites, causing more serious infections like sepsis, endocarditis, which is heart inflammation, and meningitis, which is inflammation of your meninges, which is up in your brain area or whatever. E. faecalis bacteria don't usually cause problems in healthy people, but people with underlying health conditions or a weakened immune system are more likely to get sick. And these infections often spread in hospitals. There are also, I believe, antibiotic-resistant variations of E. faecalis that can be really dangerous. Yes, the next sentence. In recent years, there's been an increase in drug-resistant E. faecalis strains. Today, many antibiotics don't work against infections caused by these bacteria. So how do you get infected by these bacteria? They can pass from person to person due to poor hygiene, etc. They are often spread through improperly cleaned catheters, dialysis ports, etc. People with organ transplants, kidney dialysis, or cancer treatment are at risk for developing infections due to immune suppression or contamination through their catheters. So basically, anytime you're on drugs that suppress your immune system, it is possible that if you get if you somehow get E. faecalis or another enterococci in a part of your body that's not your stomach, you can potentially get a bad illness. So, it sounds kind of like a double-edged sword, right? This is often how bacteria are. Usually the bacteria are good, they're doing good things in our gut, but if they get out of our gut, they're bad. So, okay. Basically, let's go back to the title, sorry. So there's this bacteria, Enterococcus, it lives in your gut normally. It's usually a good bacteria unless it gets out of your gut and into your blood or something. Now the peptidoglycan, so Enterococcus peptidoglycan remodeling promotes checkpoint inhibitor cancer immunotherapy. So basically, this paper is going to be talking about the peptidoglycan from this bacteria, so the cell wall. So just so we all remember, this is cell wall, okay? Remodeling is just kind of like a fancy term that's going to describe like how the peptidoglycan pieces might be like basically broken apart during basically when cells divide. They need to basically destroy part of their cell wall so they can split, and then the cell wall gets remade. And so that's kind of what that's referring to. And little pieces of the peptidoglycan can like get cleaved off and get secreted out into the extracellular space, which is in the gut, presumably. Moving along. So checkpoint inhibitor therapy, we talked about this. This is just an antibody, so a little Y-shaped thing that blocks the regulatory proteins. I don't need to really explain that again. So basically the abstract says the anti-tumor efficacy, so how effective cancer immunotherapy is, can correlate. So it doesn't mean it's causative, but it can correlate with the presence of certain bacterial species within the gut. And you know what? I'm just going to highlight for ease of reading because I realize, yeah. However, many of the molecular mechanisms that influence host response to immunotherapy remain elusive. So we don't really know how this works. We don't know why certain bacteria make our T cells better at killing cancer. In this study, we show that members of the bacterial genus Enterococcus improved checkpoint inhibitor immunotherapy in mouse tumor models. So as a heads up, all of these studies, all of the data is going to be done in mice, not humans. So they're saying that these Enterococci bacteria express, so they make the proteins, and then they secrete a peptidoglycan hydrolase called SagA. So let's just take apart peptidoglycan hydrolase. So when I see hydrolase, what that usually means is that it's an enzyme because of the ACE, and hydro means it's probably an enzyme that cleaves hydrogen bonds, and here it's specifically a hydrolase that cleaves hydrogen bonds in peptidoglycan, so the cell wall of bacteria. So we're talking, think about it like a little Pac-Man with like a little mouth, and it just cleaves things. And here it's cleaving hydrogen bonds on peptidoglycan, and it's called SagA. So SagA is our little Pac-Man cleaving machine. And when it cleaves peptidoglycan, it generates immune-active neuropeptides. So just think of this as like small little pieces of the peptidoglycan that stimulate the immune response. Expression of SagA in non-protective E. fecalase was sufficient to promote immunotherapy response. So what they did here is they took some Enterococci, so E. fecalase, that don't actually normally express this SagA hydrolase Pac-Man, and they engineered this non-protective bacteria to overexpress SagA. And they found that just overexpressing SagA by itself was enough to basically clear tumors. And its activity required the peptidoglycan sensor NOD2. So NOD2 is a special little receptor on immune cells that specifically binds and gets activated by little bits of the cell wall from bacteria, peptidoglycan. This is a lot of words. No, it's a lot. Notably, SagA engineered probiotics, so basically they made other bacteria express SagA, our little Pac-Man friend, or synthetic neuropeptides could also basically help with clearing the tumor. So when you guys see anti-PD-L1, just think of it as like the immunotherapy. It's the antibody that's blocking the regulatory receptors on the T cells. So when anti-PD-L1, just think of it as making the T cells better killers. So this is the therapy they're using. And then they're also adding these little peptides that bind to NOD2 on immune cells, other immune cells, that then make the T cells better at killing. Okay. Taken together, our data suggests that microbiota species with specialized peptidoglycan, cell wall, remodeling activity, and neuropeptide-based therapeutics, so copies of that peptidoglycan, those little bits, may enhance cancer immunotherapy and could be leveraged as a next generation of adjuvants. Adjuvants are just things that are added along with a main therapy to kind of help, like, basically ignite a strong immune response. When you guys are thinking about immune responses, you should kind of think of them as like, imagine pushing the first domino on a whole line of dominoes and it creates a whole bunch of like chain reactions. That's what an adjuvant does. It helps push that first domino. Okay. Yeah. Ultimately, that's the goal of this paper, is to figure out what about the bacterial cell wall is making the T cells better. Okay. So cancer immunotherapy harnesses, let's use a different color here. Cancer immunotherapy harnesses the patient's immune system to impede tumor growth and has demonstrated clinical success against a variety of solid and hematological, so blood tumors or blood cancers. In particular, so there's many different types of cancer immunotherapy, and here they're talking about antibodies that target immune checkpoint inhibitor proteins like CTLA4, PD-1, and PD-L1. And these, again, like Keytruda, have been approved to treat a number of human cancers by the FDA. Okay, so the success of checkpoint blockades relies on many factors, including the mutational burden of the malignancy. What this means is how mutated is the cancer, because if the cancer is super mutated, that means it's going to present a lot of weird peptides to the T cells. How many T cells are infiltrating the tumor? That's another determinant of how good the T cells will kill the cancer. And then signaling cues within the tumor microenvironment. This is very complicated. Basically, within a tumor, you have a lot going on. You have the cancer cells. You also have a ton of immune cells, but not all immune cells are good. Some of them are bad, and they're actually preventing T cells from doing their job. So there's a lot of crazy stuff going on, and it's very complicated. But basically, what we want here is to activate T cells. Recently, the gut microbiota has emerged as a potent factor associated with the efficacy of anti-CTLA4, anti-PD-1, and anti-PD-L1 cancer immunotherapies. When they say anti, this just means antibodies, okay? It's not like, ooh, we're against the checkpoint inhibitors. It means we're blocking these checkpoints with an antibody, like I showed before. Okay. So yeah, so basically they wanted to figure out the mechanism. All right, let's go to this part. Analysis of the commensal microbiome from human cohorts treated with different immunotherapies targeting PD-1 have found that the bacterial genus Enterococcus is enriched in patients that respond better when they're treated with this checkpoint blockade therapy. So their tumors shrink. So this is why the study here is focusing on enterococci. Okay. To evaluate specific enterococci and their mechanism of action, we used mouse tumor models and oral administration of bacteria. So they put the bacteria, the enterococci, into their drinking water. So the mice would drink and take up the bacteria through drinking in the water. So they use these special types of mice. They're called pathogen-free mice. You don't need to worry about all this. Basically, this means that these mice have a very limited number of microbes. They don't have as many microbes as like normal mice. And then they pre-treated the mice for 2 weeks with broad-spectrum antibiotic cocktails. So this contained antibiotics like ampicillin, colistin, and streptomycin to clear out their resident microbial species that may confound effects caused by enterococci. So what this means is they used antibiotics to kill all of the bacteria that normally lived in these mice so that they could then basically just give them one type of bacteria, which is the enterococci. So it's kind of creating an empty slate. Okay, so here they're doing a subcutaneous, so under the skin. That's what subcutaneous means. They were subcutaneously implanted B16 F10 melanoma cells. So this melanoma cell line is actually a very popular cell line that even I haven't used it personally, but we use it quite frequently in the cancer field. And it's actually a very difficult cancer for mice to naturally cure on their own. But anyways, this is the cancer they gave the mice, and then they treated them with anti-PD-L1, which is an antibody that's blocking the inhibitory receptor on the T cell. So if we don't remember what that is, let's go down here and I'll draw it. So you'll remember here, okay, that is really big. We'll use a different color. So here is the T cell. The T cell has its T cell receptor, or TCR. I'll just call it that. You know what? I'll just erase that. That's confusing. It has its T cell receptor, and it uses that to recognize the cancer cell. This is the cancer cell. But then the T cell is also expressing these checkpoint receptors. So PD-1, for example, and PD-1 binds to PD-L1 on other cancer cells and immune cells. So this is PD-L1. So this is a cancer. And this shuts down the T cell. So the therapy they're using here is an antibody that blocks this interaction, and it makes the T cell keep killing the cancer cell, okay? So here, what they've done is they've taken the mouse, they give the mouse antibiotics to kill all of the microbes in its gut, and then they give the mouse cancer right here, and then they start measuring the tumors. So generally after you give a mouse cancer, you have to measure the tumor to see how the tumor grows. Tumors will grow kind of like in a big upward line. They get bigger with time, obviously. And then they gave them the mice the bacteria, the Enterococcus, in their drinking water. And then they gave them anti-PD-L1. So they gave them the antibody that blocks this inhibitory receptor to make the T cells better. And then they looked at how the tumors grew. So here in this figure, what we're looking at are tumor growth curves. So over here on the left side, we're looking at tumor volume. This is how big the tumor is. And on the bottom, we're looking at the days post-injection. So remember, they inject on day 0, and then they measure the tumor after day 0, which is here. And then the line obviously gets bigger as the cancer gets bigger, right? So moving along, so if you all remember, they injected these melanoma cells under the skin of mice, and then they looked at how they grew. Now, there's several conditions that we need to look at here. First, we have the untreated condition, which is indicated by a clear box right here. So this is untreated. And as you can see, the cancer cells just like grow out really big. Then they treated some of the mice with anti-PD-L1. So this is the antibody that blocks the inhibitory receptors on T cells. So in this condition, the T cells are more activated. Yay! So this line is right here. Now I want you guys to notice something. What do you notice about these lines? This line versus the untreated line, or untreated tumors. You'll notice that there is a big difference between them that's considered significant. So basically, the tumors that were treated with the anti-PD-L1 are smaller, okay? That means that the tumors are getting killed by the T cells. This is great. This is amazing. Okay, so then they tested a couple other things. They used one of their enterococci, so the bacteria that they put in the water, they used this Enterococcus, and COM15 is just a strain name. You don't need to worry about that. EFM stands for Enterococcus faecium, which is just a very specific type of bacteria. And they put the bacteria into the drinking water alone. Now, what you'll notice about this condition, which is these green circles here, is that the tumor, it grows out pretty much the same as the untreated. There is a slight difference. It's slightly smaller, but it's not considered significant. So what this tells us is that the bacteria by themselves is not enough to control the tumor. So then what they did is they combined the bacteria with different dosages of the antibody that makes T cells better. So these are the two green solid line and green dashed line right here. So you can see that these cancers are much smaller. These tumors don't grow very well. They're being killed by T cells. And these are tumors that are in mice that, again, have drunk the bacteria, but they have also been injected with the antibody that helps T cells be more activated. So there's some type of synergy between the bacteria and between the antibody treatment that's making T cells better. Does that make sense? So that's how you read like tumor growth curve data. Okay, so this is cool. They're seeing synergy with their special bacteria and with their therapy. Okay, so then they ask a very important question. Is this effect specific to EFM or Enterococcus faecium? So then they test a whole bunch of other bacteria types. So they test E. faecalis, which is related to E. faecium, but it's slightly different. So these are all of the E. faecalis bacteria plus PDL1. And then they again test the E. faecium bacteria, which are the ones that looked good. So here's COM15 again, and those are all in green. Now you can see right away that all of the mice that were treated with different types of E. faecium strains, those tumors are really small, but all of the mice that were treated with the other type of bacteria that's somewhat related but not the same, called E. faecalis, those tumors grow out and there's like no effect with anti-PDL1. So this tells the researchers that, okay, whatever anti-tumor effect we're seeing, it's specific only to E. faecium. So this one type of bacteria. So then the researchers asked, oh, maybe these different bacteria just colonize the mice better, like these E. faecium. Maybe there's just more of them, right? And that's why they're helping control the tumor better. So then they looked at the bacterial load. So this is how many individual bacteria they got back out of the mice after they killed the mouse, they took out its intestine, and then they took all the bacteria and they basically grew them out, and they used this special math to determine how many numbers of those bacteria there are. And so you can see here they have the E. faecalis, so the EFS. These are the microbes that did not help the tumor, so these seem useless. And then they have the ones in green, which are the E. faecium, the EFM. So these are the bacteria that did help the tumor. So the tumor is smaller. And you can see that they pulled out the same number of bacteria from all the mice. So this tells the researchers that, okay, the mice were equally colonized with each of these different types of bacteria, but only the E. faecium ones were the useful types of bacteria here, okay? So then they ask, are there other bacterial species other than Enterococci that could do this similar thing? Well, actually, they look at other Enterococci, and they basically come up with a whole list. So you could see that there's E. faecalis, which is the one that doesn't work very well in blue. Let's stick to our color scheme. So you can see this one. The tumors get pretty large. There's no therapeutic effect. They look at something called EDS, which I don't remember what these ones are. I think they're more related to E. faecium, which is our good bacteria down here. So these other bacteria, they seem to have a similar therapeutic effect to our special bacteria. So they're like, oh, there's other ones, cool. And then they again check to see whether or not it's related to the number of bacteria in the mice. You can see that there's very few of these types of bacteria, but the tumor is still really well controlled. So it doesn't matter how many of the bacteria are present. It just matters if they're there. Okay. So then let's go to the next figure. So then they were like, is there an enzyme or is there something in all of these bacteria that help the tumor respond better? Is there some protein or something that's shared amongst all of them that might have anti-tumor effects? And so they do some complicated genetics that we don't need to talk about. And they find a gene called NLPCP60, blah, blah, blah. What is that? That encodes our little Pac-Man hydrolase, SagA. So SagA, remember, it's an enzyme. So the full name, oh my God, what was the full name? It was basically a peptidoglycan hydrolase. So it's a little, Rav, that scared me. Rav, hello there. How are you? Welcome in. So here's the little Pac-Man. It's a hydrolase. How do I spell that? Hydrolase. And it cleaves the sugars that make up the cell wall. So this is cell wall, or peptidoglycan, of bacteria. And it turns them into little bitty pieces. And these little bitty pieces are called peptides. Don't ask me why they came up with such a stupid name. They could have called them like peppies, short for like peptidoglycan. It's like little peppies. Anyways, the hydrolase SagA cuts up the cell wall into little tiny peptides. All right, that's cute, fun. So then they asked, oh my God, are there other SagA proteins that are present in other enterococci species? And so they do a whole bunch more genetics. They look at things like lactobacillus, other bacteria, to see if they also make SagA, because then they could apply this to other bacteria that are inside of humans, right? Basically, they want to know, are there bacteria that colonize human stomachs that have this special enzyme? Now, at this point, they haven't even gotten into whether or not it's this SagA hydrolase that's helping the tumors get cleared better by T cells. So they still need to do that. But anyways, they find that there's some lactobacillus that express this SagA protein, and then there's E. faecalis, which expresses it, and then E. faecalis, there's actually not detected. ND means not detected. So that means there's probably no E. faecalis that actually makes SagA, which makes sense because if we go back here, remember that E. faecalis is our shitty bacteria? You add E. faecalis to the drinking water of the mice and the tumors keep growing really big. So E. faecalis are shitty bacteria. Okay, sorry. And they also find that E. faecalis they make no SagA. But our good bacteria, E. faecium and other enterococcus species, that actually did control the tumor back here. So these guys, they all make, and then also these guys, they all make the SagA protein, okay? Super cool. So they're like our awesome bacteria. They're not shitty like E. faecalis. All right. How are you guys doing? So now that they've like looked at the genetics of the bacteria, so they've looked at the DNA, they're now like, okay, do these bacteria actually make the protein SagA? Do they make our little Pac-Man friend? So now they're looking for the protein. And what they do is they basically grind up the bacteria. Well, actually, here they're looking at secreted proteins. So they basically grow the bacteria in broth. Then they like remove just the broth, not the bacteria. They spin the bacteria down probably. I'm not sure what they did. But they just take the proteins that these bacteria secreted, which they sometimes will secrete SagA to go around and like cleave, like it has to be secreted to then go and like cleave the cell wall of like either itself or other bacteria. And they find that, let's look at this column first, the one I'm circling in green. They find that E. faecium, so this is our good bacteria, it expresses a lot of the SagA protein. So what we're looking at is something called a Western blot. So a Western blot is just a technique that takes a bunch of proteins and runs them on a gel, and the proteins get separated based on size. So you'll see up here, this is this thing called KDA, stands for kilodalton. So kilodalton is just a measurement of protein size. And the bigger proteins are at the top, and the smaller proteins, so big at the top, small proteins at the bottom. The big proteins at the top are at the top because they're so big, they travel through the gel slower. But the small proteins, they can travel through the gel faster. So they're going to be down here. And then what they do to kind of visualize, because there's many different types of proteins, right? To visualize the specific SagA protein, they use an antibody that recognizes SagA. So that's why there's this little alpha sign here That stands for anti or antibody. You could think of it that way. They use an antibody that specifically binds to the SagA protein, and then they use like this little label, and basically it makes this black stain. If there's a SagA protein, if SagA is present, it'll make a black stain. Now, they used a negative control over here. I believe this is a bacteria that normally does not make SagA, and you can see that there's none. So this is the negative control, no SagA protein. And then they look at, oh, this is important. Let me erase all of this. So they also look at E. faecalis. If you'll remember, E. faecalis is our shitty bacteria. If you remember the tumor growth curves, the tumors just grow out if you add E. faecalis to their drinking water because they don't make SagA. And we can confirm this here because look, there's no SagA. Oh my God, it's beginning to make sense. Maybe. This still hasn't confirmed anything about whether or not SagA is actually stimulating the immune response. We haven't gotten that far. Sorry. And then what they do, so they do a little bit of protein engineering. So what this means, oh my God, I'm trying to touch this. So, all right. So over here in this condition in orange, we see that we have EFS again, so shitty bacteria, but oh my God, it's now expressing SagA. And you'll see in the title, it says EFS SagA. What this means is that they basically engineered the shitty bacteria to overexpress SagA artificially. So normally the E. faecalis does not make SagA, but they added the SagA gene into the bacterial genome so that it would then make SagA protein. And you can see here they're confirming that indeed this bacteria makes a shit ton of SagA, which is great. This is good. And then they look at all of their other enterococci species that also had anti-tumor effects, and they see they also make SagA proteins. So they're basically confirming, okay, SagA is a protein that is both present in the DNA, so the genome of the bacteria, and it's also made as a protein in the bacteria that helped control the tumor really well. So maybe SagA is important. It's Clover. Thank you for that follow. Welcome in. It's great to see you here, by the way. Thanks for joining the stream. It is a lot to take in, and I'm sure that at this point you guys are just like hanging in, like, oh my God, I don't know what's happening. Don't worry, that's me. Immunology is one of the hardest fields to understand as a outsider or just like anyone. It's a really hard field. So the fact that you guys are even like grasping just a little bit of what I'm saying is really awesome. Okay. Oh, thank you for the hydrate. I'll hydrate with water. I also have tons of coffee. Geoff, welcome to the stream. Thank you for that follow. I hope you are doing fantastic today. Also, I see Donuts entered the chat. Donuts, you're at TwitchCon right now, aren't you? How's TwitchCon? I hope it's well. I see some more questions. Are these strains available? Pixel Fiend. I'm sure that we could get these strains. So first of all, the non-engineered strains, so like EFM and E. faecalis, these are available. Yes. Now, by no means would I suggest you ingest these. First of all, your body already likely has E. faecium and E. faecalis, the shitty bacteria. It likely already has some of these enterococcus species living in your gut. And also, just because these results here suggest that these bacteria have an anti-cancer effect when combined with checkpoint blockade therapy, it does not mean that you should go ingest them, because it might not actually, we haven't confirmed yet in controlled clinical trials that this is true. Although I do have to check that. This paper was published a few years ago, so by now there could be a clinical trial that's active. That's something I should check. But anyways, yeah. I'm sure if you wanted to do your own studies in mice or whatever, you can go purchase these bacteria on the magical lab stores where you can purchase bacteria and DNA and fun things. You can also isolate them from your own poop if you're interested, but you have to kind of know what you're doing so you don't isolate something dangerous. Okay, so continuing on, if you guys are just joining, the paper we're discussing is pinned in chat. We're basically talking about how these certain Enterococcus bacteria, they have an enzyme that basically makes a small peptide that then activates the immune response and makes T cells kill cancer better. Okay, so in this figure, in this Western blot, they're just confirming that the bacteria that's really good at killing cancer does indeed express this SagA protein, and the bacteria that does not do great at killing cancer, it does not express this protein, but they can use cool protein or genetics to basically make the bad bacteria express this SagA special enzyme. Okay, and this is going to come up later because then they're going to test whether or not this new engineered strain of E. faecalis with SagA can actually help the tumor, like clear the tumor. If that, basically that would suggest that SagA is sufficient for causing this anti-tumor effect. So can you get the SagA protein without the bacteria? So SagA could be made by itself. The only thing is there's another way to get to kind of bypass bacteria even better, and that is to simply make synthetic copies of GMDP. So remember how I told you there are some companies that are really interested in using the cleaved portion of the cell wall as a therapeutic? You can just make this in the lab, this GMDP. In that case, you don't need the bacteria. You don't need SagA. You could just make this. Okay? But at this point, during this study, they still weren't sure if GMDP was actually causing the anti-tumor effects. They're just kind of looking at this interesting protein, SagA, and what it does. It cleaves cell wall and it makes GMDP. Okay, cool. Now they need to see if that actually helps cure the cancer, right? But yeah, to answer your question, metkabutane, there is actually some companies that are making the little cleaved versions of basically similar to GMDP as a cancer thing. I actually don't know what GMDP stands for, and I love your take on it. Good immune defense peptide. I love that. That's freaking awesome. Okay, so now in this figure that looks really complicated, don't worry, we're going to zoom in and look at it close up. This figure is all about answering the question, is SagA hydrolase sufficient for tumor control? So remember, they've identified SagA as being a protein that is made by the specific types of Enterococcus that are really good at killing the tumor. But can SagA by itself basically be enough to kill the tumor? So what they're doing here in this first experiment, they're again using the B16F10 melanoma cell line, so melanoma, skin cancer. They basically implant the skin cancer cells under the skin of the mouse, and then it grows into a little bulb, and then they measure the size of the tumor here. So here in this figure A, they have the tumor volume here. So the bigger the tumor, the bigger the number, right? Pretty simple. Oops. God. And then down here they have the days post-injection. So this is just following the growth of the tumor over time. So again, they have an untreated control. That's this guy. It grows up. The tumor gets really big. Then they have anti-PD-L1. Now, very interesting to me here is that anti-PD-L1 alone, this thing, is not as, like, the tumors are still pretty large compared to what we saw back in the first figure. But anyways, basically this is showing that anti-PD-L1 by itself is shitty. Okay. Then, as a positive control, they add anti-PD-L1 with E. faecium, so EFM, that's our good bacteria. You can see down here, the tumors are really small. That's good. This is what they saw back in the very first figure. And then as a control, they also take E. faecalis, EFS, the shitty bacteria. You can see that the tumors get really big compared to the E. faecium. There's a huge difference here. All right, this is all corroborating what they've seen before. But then they do something really interesting. They take the shitty bacteria they've like they've basically given the DNA to make SagA to this shitty EFS bacteria. So now the EFS bacteria can make SagA, which they confirmed in this Western blot right here. This is SagA made by that shitty bacteria that's engineered to express it. And they give that special engineered bacteria to the mice, and oh my God, what do you guys see? The orange triangles. That's the EFS SagA bacteria. So this is showing that just by adding SagA to a bacteria that normally doesn't express it, that's enough to get tumor control. So this experiment is clearly showing us that the SagA hydrolase, the little Pac-Man, is sufficient for eliciting this tumor control. Wow, super cool, right? It's just the one you pull out of your ass the last minute. Trust me, I do a lot of experiments that I just pull out of my ass, okay? I'm working through my 3rd cup. Okay, I am on my second. Me and Caffeinated Linux made a bunch of coffee earlier, and we were streaming a coffee tasting, and we're still consuming the coffee. I'm going to be up. Major Magruder, hello, welcome to stream. How are you? Pac-Man for the win. Yep. All right. So this was a really cool experiment. Now they wanted to show that this SagA protein and these bacteria can also control other types of tumors. So now in figure B, they're using something called an MCA 205 tumor. So MCA just stands for methylchloranthine. This is a type of carcinogen that when injected into the skin of a mouse can cause a muscle tumor to form. Fun thing, I've actually injected a ton of MCA into my mice, and now they have tumors, and it's really cool. Basically, what this MCA tumor is they took the tumor that formed after they injected the MCA, the carcinogen, they took it out of the mouse, and then they cultured it in like the lab. And basically, because these are cancer cells, they can replicate on their own. They're immortalized. And they made a cell line called MCA 205. 205 probably refers to like the clone, because then they have to like sort these cells out and make clones of them. So anyways, I erased that. Too bad. So they're using this special muscle tumor. So here we'll just write muscle tumor. Now the interesting thing is they're still injecting the muscle tumor just underneath the skin. So just imagine that this is mouse skin, and then they just inject the tumor into it, and then the tumor grows out in a little bulge. So here you can see again that the untreated tumors, they grow out really big. The anti-PD, now they're using anti-PD-1, which I'm kind of confused why they switched. Basically, anti-PD-1 or anti-PD-L1 will block the same inhibitory interaction that's happening with T cells. So either of these therapies should theoretically activate the T cells. So that's fine. But they see that either untreated in anti-PD-1 alone, basically the tumors don't respond, they grow out really big. They then use our good bacteria, E. faecium, and you can see down here the tumors are controlled. Awesome. And then they also use our shitty bacteria, E. faecalis. You can see these tumors grow out. All right, nothing new here. And then they use the E. faecalis that has SagA added, so our special little Pac-Man enzyme. And you can see these tumors are really well controlled, just like E. faecium. And this, again, is supporting the claim that SagA is sufficient for tumor control. They do this one more time in a different tumor cell line. This tumor cell line is called MC38. So basically, this is a cancer that originated in the colon of a mouse, and then they took it out and they made a cell line out of it, and it's called MC38. So they use this tumor cell line, and they again do the same groups. They have untreated, tumor grows out really big. This time they're adding anti-CTLA-4, which again, I'm not sure why they're switching up the therapies. Maybe they just want to see whether or not each of these different types of checkpoint blockade therapies synergizes with the bacteria. I'm not sure. But they use anti-CTLA-4, which again, if you remember, is another receptor on the T cell that, if it binds to its ligand, can inhibit the T cell. But if you block CTLA-4, then the T cell is awesome and it works really well. We can also see that when we use the shitty bacteria, EFS, it basically looks the same as the anti-CTLA4 alone. So these two look the same, which basically tells us that E. faecalis, our shitty bacteria, is not doing anything here. When we use our good bacteria with anti-CTLA4, though, we see really good tumor clearance. The tumors are really small. And then when we add our E. faecalis, the shitty bacteria, with SagA, we also see really good tumor clearance. So again, this is just showing us that SagA is helping clear the tumor. All right, so moving back along, so now we have confirmed that SagA, this Pac-Man-like hydrolase, which is cleaving hydrogen bonds of the bacterial cell wall and making these tiny little bits of cell wall, that alone is enough to trigger this really awesome anti-tumor response when combined with anti-PD-L1 or a checkpoint blockade therapy. Okay, cool. So now they ask, how is this happening? What is SagA doing? And so this is where they start looking at the immune cells, because SagA by itself, like if you think about it, how would a small peptide kill a cancer cell? That does not make sense. So what must be happening is that the little peptide is somehow being bound by an immune cell that actually has the ability to kill cancer, right? So now they're looking at the immune cells to see if maybe in the SagA treated mice, or the mice that have the bacteria with SagA, maybe there's more immune cells in the tumor. So what they do is they take the tumors out of the mice, they grind the tumors up into individual cells, and then they stain the cells for different immune cell markers. Okay, anyways, so all right. We've established that in tumors with our SagA hydrolase, there's more T cells, which is good. These are the cells that would typically kill the cancer. And if there's more of them in the tumor, then this suggests that maybe they're the cells that are killing the tumor. And maybe SagA is somehow triggering the recruitment of T cells into the tumor. Then they looked at something. So they looked at FOXP3 positive cells. Now, FOXP3 positive cells are the Treg cells. These are the T regulatory cells. These Tregs, they actually shut down T cells that might be killing the cancer. So they looked to see if there were more Tregs in the tumor, and guess what? With the SagA treated mice, there was actually no difference in the number of Tregs. So SagA isn't really causing more Tregs to be in the tumor. That's good, because then if there were more Tregs in the tumor, you could imagine they'd be shutting down the good T cells, like the T cells that are killing the tumor. Now they're looking at CD8 positive T cells. So CD8 positive cells, these are what we call cytotoxic T cells, and they express this protein called CD8. And cytotoxic T cells, cyto means cell, and toxic obviously means bad. So cell toxic T cells, that means these are the T cells that are killing the tumor cells. So these are special. You can see that in our shitty tumor, EFS, so now they're looking at the percent of CD3 positive cells. So they're looking at what percentage of all T cells are CD8 positive. We're not looking at total number anymore. And you can see here that there's only about 40-50% of the CD3 T cells that are actually cytotoxic. But in the SagA expressing bacteria, you can see that now that's shifted up to about 60% of the T cells. So there's this small increase in the cytotoxic T cells. That's cool. Then they look at the cytotoxic T cells that are expressing something called granzyme B. All right, you guys are really learning a lot of immunology today. You should all pat yourselves on the back. Granzyme B is basically this little, it's an enzyme. That's why it's called granzyme. It's an enzyme that actually initiates apoptosis. Okay? We're going to just say initiates apoptosis. And as you guys know from earlier, from our awesome apoptosis videos, apoptosis is cell death. So cytotoxic T cells, what they do, is they like basically create a hole in their target cell, and then they like shoot granzyme B into the target cell, and then granzyme B like goes into the target cell and it cleaves these things called caspases that then result in apoptosis. And then the target cell, which is the tumor in this case, explodes and dies, and it's really fun. So they were looking at cytotoxic T cells that were expressing granzyme B. Normally, if cytotoxic T cells are activated and really good at killing things and they're uninhibited, they'll express a lot of granzyme B inside of them because they're making it all the time so they can shoot it out into their target cell. You could think of it as like bullets or something. So you could think of it as like T cell ammo. So the mice that got the bacteria with SagA, they have T cells that have more of this special T cell ammo, okay? So that's really cool to see that. Now, finally, they ask whether or not the T cells that are in the tumor are actually specific to the tumor. So they can do this. This is actually a complicated experiment, so I'm just going to maybe pass it up. Basically, they use a special antibody that recognizes a T cell receptor on the CD8 T cell. So this is the cytotoxic T cell. And this T cell has a receptor. So this is my really shitty T cell over here. T cell. This T cell has a T cell receptor. This is not an antibody. This is a receptor. We have an alpha chain and a beta chain because remember, T cell receptors have 2 chains. And this T cell receptor recognizes only one single type of protein. So this is actually true of all T cell receptors. They only recognize one type of protein, but every T cell recognizes a different type of protein. So this T cell recognizes the OVA protein. OVA stands for ovalbumin, which is just, I believe it comes from like chickens. It's like a artificial protein that you wouldn't normally find in a mouse. So this is an OVA-specific T cell. What's the point of this? Well, what they did in this experiment specifically, in eye, is they injected a tumor into the mouse that expresses this OVA protein, and then they looked for T cells that recognize the OVA protein. So then they looked for pink T cells. Well, just because we're using pink here. They looked for any T cell that has this T cell receptor. And this tells them that these T cells are very specific. Like if there's more of these T cells, it means they've become activated specifically to the tumor, and then they've made copies of themselves, because that's what T cells do. When a T cell recognizes its target, it gets activated, and then it makes tons of copies of itself. So you can see that here in this graph. You can see in the SagA tumor, which we know, or in the SagA treated mice, which we know have a better response to the cancer, you can see that there's way more of these OVA-specific T cells, which means they've made more copies of themselves, versus in the shitty bacteria-treated mice, okay? What they're now asking is how, like, is the little GMDP peptide that SagA makes, is that, it's triggering the immune system, obviously, because they see more immune cells, more T cells, more tumor-specific T cells, etc. But how is it detected? Now, they already had a hypothesis. They already kind of knew that GMDP can be recognized by this immune receptor called NOD2. Okay, this is an immune receptor. And it's found on it's actually not found on T cells. It's found on these other types of immune cells. So now this is where mouse genetics gets really awesome. What they do is they knock out NOD in their mice. So these mice, they can't make the NOD receptor. And that's signified by NOD2 minus. That means that the mouse lacks both of the gene copies or alleles to make NOD2. So it can't make the protein at all. And then they use a control. They use the heterozygotes. So heterozygotes, that basically means that these mice up here, they have a NOD2 plus minus. This means these mice are only lacking one of the functional alleles for making NOD2, but they still have one functional copy. So they can still make NOD2. So these, they make NOD2, they're NOD2 positive, and then the ones down here are NOD2 negative. They have no NOD2 protein. So now what they're doing is they're using their shitty bacteria as a negative control, and they're also using the shitty bacteria that overexpresses SagA, so the orange one. And they're combining this with anti-PD-L1 in their sarcoma, or in their melanoma cell line, or melanoma tumors. So let's first look at anti-PD-L1 with shitty bacteria, E. faecalis, and then in mice that are NOD2 positive. And they've done this experiment before, like with the basically with mice that express NOD2, and you can see that the tumors grow out. Tumors are not well controlled. The tumor growth curve shoots up. Then what they do is they take E. faecalis plus anti-PD-L1, and they do the experiment. They add, they basically give the bacteria to NOD2 deficient mice, so mice that don't make NOD2. And here you can see again the tumors grow out really big. You can't really see the curve because it's being covered by the orange triangle. But if I zoom in, you can see there's a blue line underneath, like right here. So basically, this is also ineffective. But again, they're still using the shitty bacteria, so we don't expect an anti-tumor response. Then what they do is they take anti-PD-L1, and then they take the shitty bacteria plus SagA, and they do it in a mouse that expresses NOD2. So this one here, this is NOD2 positive, and we see what we saw before. We see that the tumors are really well controlled. But then they do the same thing with the special SagA expressing bacteria, but they do it in mice that don't have the NOD2 receptor. And here, this is the interesting part, we see the tumor grow out, this is the empty orange triangle right here. So these two are basically the same conditions, except they've knocked out NOD2. And you can see there's a vastly different phenotype. When they knock out NOD2, tumors escape, which means NOD2 is necessary for eliciting and helping activate the anti-tumor immune response. So somehow these non-T cells that express NOD2, they must produce something that activates T cells. And they found that out by knocking out NOD2. And NOD2, again, it's the receptor that binds to GMDP, which is the tiny little peptide that gets cleaved by SagA. Okay, so that's how it's related back to SagA. SagA just makes the peptide that's bound by NOD2. Okay, also, if you're wondering how long this paper is, we're basically about to do the last figure. So we're almost done. I know this has been a lot. Okay, so then they ask, do the bacteria have to secrete SagA to trigger an immune response? So usually SagA is secreted by the bacteria normally. Remember, they did that Western blot experiment and they just used secreted SagA. But the other question they have is do the bacteria have to secrete it out into the, into the lumen or into the gut? And also, what happens if they knock out the hydrolase activity of SagA? So what if they make SagA not able to cleave? So they basically take Pac-Man's ability to cleave away. So now it's just frozen. It can't cleave anymore. So they make two different types of SagA proteins, and I think they put these into a different type of bacteria. So the LL right here, this is actually a lactobacillus strain. This is a bacteria that normally doesn't make SagA, because you can see that if you just take this lactobacillus by itself and you look for secreted protein, it does not make SagA. There's no SagA here, right? So then they engineer this lactobacillus with the normal SagA, right? The one that should be functional. And they see, okay, there's SagA that's secreted. That's awesome. Then they make a SagA mutant called C44-3A, and this means that it has no more cleaving activity. So this is a non-functional, kind of like a dud. And then they make a SagA that can't be secreted. So this SagA, you could imagine that it's just stuck in the bacteria. So it can't be excreted out into the world. And so then they kind of confirm that these new bacterial strains work as anticipated. So in this panel here, they're looking at secreted protein. You can see that when they knock out secretion, and they're like delta SS, that means delta just means like knockout. So they knocked out the secretion function. You can see that there's very little SagA present. And then when they look at the pellet, this is just like the actual bacteria. You can see that there's a lot more SagA present in the bacteria because the SagA is just stuck in the bacteria. Okay, so that's just to confirm that these new mutant bacteria are working functionally. And so anyways, they take these new mutant strains of lactobacillus and they do their cancer growth curves again. So again, they're looking at tumor size, how it increases over time, and then they're looking at that over days. And so here they have their anti-PD-L1. So again, anti-PD-L1 is the therapy that makes T cells better, and they add it with the lactobacillus, just like wild-type lactobacillus that don't make SagA. You can see that tumors are not controlled. All right, that's expected. They don't make SagA. Then they do the same experiment. They give the mice anti-PD-L1 and they give them the lactobacillus that makes wild-type SagA. So this is like the normal type of SagA that the other good bacteria made. And you can see that the tumor is really well controlled. So the tumors are really small, which means that the T cells are able to kill the tumor better. And then they use the mutant bacteria that doesn't have, basically this is the lactobacillus with the SagA that it doesn't have the hydrolase activity anymore, okay? So it basically is stuck. It can't like cleave the peptidoglycan cell walls, and it can't make the GMDP peptide anymore. So this is like, basically this is a broken SagA. It's non-functional. And when they break SagA, you can see that the tumors grow out and there's no tumor control. Okay. So it's basically equivalent to when you don't have SagA present. And then they also try the bacteria that has the SagA that can't be secreted. So again, they were asking here, like, does SagA have to be secreted to cleave like other cell walls, or if it's just retained within the bacteria itself, is that enough to make the peptides that will elicit the tumor control? And so when SagA cannot be secreted, this is this pink triangle right here, you can see that the tumor is it's like somewhat controlled. Like the tumor is much smaller than when you break the catalytic function or like the enzyme function of it. So you can see that basically this tells us that SagA does not need to be secreted to still elicit an anti-tumor response. So it can cleave its own bacteria's cell wall, like the one that is the bacteria that's making it, and that's enough to make like little peptides that then go out and activate the immune system. So now we're finally at the last figure, guys. Oh my God, what a journey. So we're at the last figure, and now they're finally just looking at the peptides themselves, the GMDP, so the products of SagA cleavage. Because if you'll remember, SagA, it's like this little Pac-Man. It cleaves the cell wall into tiny little pieces. And so now, and this is the chemical structure of those tiny little pieces. Now they have two versions, one that's NOD2 inactive and one that's NOD2 active. What this means is that so I don't actually know what it entirely means, but I'm guessing that this version, when it binds to NOD2, it doesn't activate NOD2. Like it can bind NOD, the receptor, the immune receptor, but it doesn't activate it. Whereas this green one, this one is active. When it binds NOD2, then it activates the immune cell, which then starts this whole domino effect that somehow results in the T cells being better cancer killers. So they take just these peptides. So now we're not even using bacteria anymore. No more bacteria in the water, okay? Now we're just giving the mice cancer, and then we're giving them the two different types of peptides. We're either giving them the inactive one, so when it binds NOD2, nothing happens, or we're giving them the active one.
Little coffee bean. Welcome to the caffeine club.
Iron, welcome in. Thanks for that resub. Oh my God, 11 months. How are you? we're just doing a deep dive on a cancer paper, and if you're interested, it is linked in chat. It's one of the pinned messages. You can open the paper yourself. You can find this figure. This is figure 4. For those of you who are like trying to follow, like, where the F are we? We are at figure 4, which is basically the end of the paper. So you can pop on down to figure 4 so you can see these graphs for yourself, like up close. SagA being a protein that is produced in certain gut microbes, and saggy cleavage can be unnecessary. Yeah, you can remember SagA and its function as saggy cleavage. Because it's a protein that cleaves. Okay, sorry. I'm sorry. That was dumb. That was funny, though. I couldn't help myself. All right, so now we're just looking at the peptides that are cleaved by SagA. And so when we add the inactive ones, so now they're just injecting the peptides straight into the mouse. They inject them into their peritoneum. So what they do is, like, imagine someone just takes a needle and they stick it in your stomach. That's what they're doing to the mice. So they're just injecting it into, like, the cavity, like the organ cavity of the mouse. And then the SagA peptide just like, they go somewhere and they activate the immune cells that have the NOD2 receptor. So, okay, the blue curve here, this is the inactive version of the peptide. And you can see when they inject the inactive version of the peptide, nothing happens. It binds NOD2, but it can't activate NOD2. So there's no anti-tumor response. But if they inject the active version, so the green one, you can see that the tumors are really small. Like, the tumors like try to grow and then they're cleared by the T cells. That's dope. Now they're going to do some fancy technique called single cell RNA sequencing. So now they're looking at every single cell that they pull out of the tumor, and what they want to do is they want to figure out which cells first off are expressing the NOD2 receptor in the tumor microenvironment, and then, like, what are some of the, like, genes that are turned on in each of the immune cells that are located in the tumor. And so basically they use this technique where they take each cell and they look at the RNA transcripts that are made in that cell. Now, what the fuck is RNA? All right, we're going to go back and do some basic biology. So I'm sure you guys are all aware that we have DNA in our body, and our DNA has these little things called genes. Now, I'm sure you guys are like, that's not DNA. DNA is a helix. Okay, I'm just showing you, like, a DNA map, which is basically we look at DNA in, like, strands, and then we look at, like, where the genes are located on the DNA. So DNA, it's retained in every single, like, the same DNA is in every single one of your cells. They have the same copies of DNA that can make everything. However, not every cell makes everything, because there's a whole bunch of, like, controls in the cell that are cell type specific that will turn genes on and off. Now, let's say that a gene is turned on. So usually in front of a gene, there's something called a promoter. The promoter is controlled by a number of different things. And when that promoter is turned on, kind of like a light switch, there's this cell machinery that will convert the DNA gene. So basically the gene is just a fragment of DNA that encodes a specific, like, complementary piece of RNA. So when the promoter is turned on, what happens is there's a little piece of RNA that's made. And depending on how strong the promoter is, you can have tons and tons of copies of this RNA that are made. And then what we can do is we can take a single cell and we can measure, like, pretty precisely how many copies of the RNA are made. We can measure that. And so what they're doing with this single cell RNA sequencing is they're measuring how many copies of RNA are made. Now, you might be wondering, okay, why does RNA matter? Well, it matters because RNA gets converted into proteins. So each little RNA becomes its own little protein, and proteins are the things that actually do the work in the cell. Like, your cell is built out of proteins, okay? So the RNA, you could think of it as like so the DNA is a blueprint, the RNA is like a copy, and then the protein is like the functional, like, I don't know if you guys are engineers, but have you ever used what is that program called? AutoCAD. Have you ever like used programs to make like a blueprint for something, and then you send it to your 3D printer, and then your 3D printer prints out whatever design you designed on, I don't know, AutoCAD? Basically, you could think of like this whole process right here as like the blueprint being sent to the 3D printer, and then the 3D printer is printing out like the actual thing that actually does the work. So the protein, so that's how RNA translates to protein. So scientists will sometimes measure the numbers of RNA that a cell makes, and that kind of correlates to the amount of protein the cells are making. You might ask, well, why don't they just measure the protein? That's actually technically challenging, and it's much easier to measure the amount of RNA. We have good techniques for that. So what you're seeing in this figure E here is the percentage of RNA that's being made for, that's specific to each of these categories down here. So there's basically RNA that's associated with active T cells. This could basically be a whole bunch of different types of RNAs that are associated with different genes that are turned on only in active T cells, and so they're only looking at that RNA. And what the big takeaway is that when they add the inactive version of MDP, which is that little peptide that is made by SagA, which is made by Enterococcus, if they look at the inactive version, you can see there's just a small little amount of these active T cell genes that are turned on. But then, if they use the active version of the peptide, you can see there's now a significant increase in the amount of RNA that's being made for active T cells. Does that make sense? Oh yeah, CAD. Oh my God, hello Mav, welcome in. I hope you're doing well. Welcome into the stream. Can we remove, wait, why isn't this scrolling down? Can we remove the DNA that causes diarrhea? I'm tired of it running in my genes. Leb, that was good. That was real good. Guys, you should check out Leb's channel. He's a lovely streamer and he makes tons of puns. It's like you have the whole book, but the teacher tells you only read chapters 3 to 5. Yeah, that's a great analogy to how DNA is turned on or off. I'll print out a kettle like Caff's. All right, good luck with that. I hope it doesn't melt. Ribonucleic acid? Yes, that's what RNA stands for. I remember seeing this really hippie video about how ribosomes copy proteins, and this dude kept shouting, tRNA. Okay, so first of all, ribosomes make protein. What ribosomes do is, so when the RNA is getting made into protein, it's actually made by the ribosomes. So the ribosomes, they'll basically bind to the RNA, and then they'll like add on new amino acids to the corresponding codons in the RNA. So let's say you have, so basically a codon is 3 nucleic acids, and nucleic acids are like A, C, T, and G. And then in RNA, it's basically complementary to that. But basically a codon is made up of 3 nucleic acids, and each codon corresponds to 1 amino acid, and amino acids are these like chemical structures that make up proteins. So the ribosomes, they'll basically read the RNA, and then they'll take the different types of amino acids that correspond to the codons, and they'll like stick them together and make a protein. Something like that. So you could think of the ribosome as the 3D printer, I guess. Okay, so yeah, basically we're looking at RNA here, not protein, and we can see that there's that in our active, so in the green bars are the active peptides, and we can see that overall there's like more Like here we have monocytes, like a type of monocyte. Monocytes are not always good for cancer, but I think these ones might be because they said that there was a class of monocytes in the cancer that was expressing a lot of NOD2. And remember, NOD2 is what's binding to the peptide MDP, and MDP-LD is the active isoform of the peptide. And so they see that when they add the active form of the peptide, there's like more of these ones, which I don't know, it could happen before or after. It's not really clear. Felon, good night. Thank you for hanging out. I hope this was somewhat educational to you, and I appreciate you being here. But anyways, I found this data, like, it's okay. It's like kind of correlative. It's not really showing actual cells increasing. They're just looking at like the RNA transcripts. They also show that suppressive cells like macrophages, so sometimes macrophages can be bad. There's less macrophages in the mice that are given the peptide that activates NOD2. And remember, when NOD2 is activated, you get better cancer control. So there's less macrophages in the tumors that are better controlled. All right. And then the rest of this looks like bullshit. Just kidding. The rest of this is just more like RNA data. They're looking at different, basically these are different inflammatory genes that are turned on. And they say that there's basically more inflammatory genes turned on when they add the active MDP peptide. And then this is just a schematic of how this is all working, or how they think it's working. And this is like the last thing, and then I'll be done with this paper. So here, remember MDP, which is similar but not quite the same as GMDP. So if you'll remember, MDP is synthetic, but it has a similar structure to GMDP. This is made by SagA from the Enterococcus. So they basically show that both of these proteins, MDP and GMDP, they're known to activate the NOD2 receptor. NOD2 then it gets activated, and it turns on this other protein called RIPK2, which then turns on another protein called TAC1, and that turns on NF-kappa B. NF-kappa B is super important. It is a protein that it's actually a transcription factor. Transcription factor is one of those light switches that turns on, basically turns on the promoter for a whole bunch of genes that are inflammatory. So NF-kappa B is the light switch. When a signaling event outside of the cell basically results in NF-kappa B being activated, then you get genes getting turned on that make these pro-inflammatory proteins like IL-1 beta and NLRP3. MAPK is another transcription factor, I think. But anyways, so basically they're showing that the little peptides that are made by SagA cleaving the cell wall, that is activating this inflammatory pathway that eventually results in inflammatory genes being turned on. And that's kind of like their final figure. There's a couple takeaways that I'll go over. Hoju, hello, you just walked into hot genes. Hoju, we are reading a really cool scientific paper about how it's basically studying the mechanism of how different gut microbes can actually help promote an anti-tumor or anti-cancer response by resulting in more activated T cells, and how it synergizes with checkpoint blockade. Okay, so some final takeaways. So their takeaway is that our data, here, let's color this in. Our data indicates that enterococci, which are the bacteria that normally live in your gut, with distinctive NLPC P60 peptidoglycan hydrolase activity, so SagA, all of this just means SagA, can generate NOD2 active muropeptides. Again, muropeptides, what are they? GMDP, and then the synthetic version is MDP. So SagA makes GMDP, and this modulates the efficacy of checkpoint blockade immunotherapy in vivo, which just means in the mouse. Modulating the efficacy, what that means is it's helping make checkpoint blockade immunotherapy better because it's somehow activating more T cells. All right. And then they continue to say, although enriched bacteria in responding patients, so they're the patients that respond better to checkpoint blockade therapy, do not correlate well regarding phylogeny. What this means is that the bacteria that are enriched in the patients that have responded well to checkpoint blockade therapy, they don't always include enterococci that have the SagA protein. But they say there could be specific enterococci and other microbiota species with privileged cell wall composition and remodeling activity that could provide functional indicators of therapeutic efficacy, blah, blah, blah. When I read that sentence, my brain cells died. It's a very vague sentence, and I'm not quite sure what they're trying to get at. Let me think about this. Although enriched bacteria in responding patients do not correlate well with in regard to phylogeny, specific enterococci and other microbiota species with privileged cell wall composition and remodeling activity. Yeah, I still don't understand it. Whatever. So then they go on to say that our results demonstrate the prevalence of conserved SagA-like enzymes throughout human-associated Enterococcus species. So remember in figure 2, they did find there were other SagA-like enzymes in other Enterococci species in humans. And maybe this indicates that there's also production of immune active neuropeptides. But again, they did not test in this paper whether or not human microbiomes produce the SagA enzyme or neuropeptides. Beyond enterococci, the detection of potential NLPC P60 orthologs, so proteins that are or basically genes that are similar in other genres of bacteria like lactobacillus, which have also been identified in immunotherapy responsive patients, may provide additional specific microbiota correlations to predict and improve patient outcomes. So what they're saying here is that what they would eventually like to be able to do is take a fecal sample from a cancer patient, then send that sample in for sequencing. Basically, they can sequence and determine the abundance of different microbial species in that fecal sample. And then they can determine if there's like the special lactobacillus species that make SagA, or if there's the special enterococcus species that make SagA, then that could be a predictive marker that that patient will respond really well if they give them checkpoint blockade therapy. Because sometimes it's like, why would you subject someone to checkpoint blockade therapy if it's not going to actually help them? Which, to be honest, because checkpoint blockade therapy does have some bad side effects, like it could actually result in some autoimmune inflammation that happens because you're activating all the T cells, and some of those T cells might recognize tissue that is healthy and attack it. This does happen in some cases. And so they want to make sure that they can just predict the responder, and they want to be able to do that by sequencing the microbes in the patient's poop, which is, again, we're coming back to poop. Also, IO Relic, welcome to the stream. I hope you're doing well. Thanks for joining us. We're actually at the end of our paper, but if you're interested in reading it on your own, you can check it out on the pinned link right up here in the chat, and you can get it for free because this is an open access paper, which is great. In addition, they say there's been recent genetic analyses of bifidobacterium, which is another type of bacteria that synergizes with PD-1 blockade in patients. And these bacteria showed an enrichment of peptidoglycan biosynthetic genes. That's pretty vague. They're saying, well, they can make peptidoglycan, but so maybe SagA produced by enterococci can cleave the peptidoglycan that these bifidobacteria make. I don't know. This is kind of vague. Hi there, okay Tom. Welcome to stream. How are you? Got some sag going on here from that, and it all comes from Tara being cockside. Leb, oh my god. Okay, that's stretching it. You're getting further and further away from being helpful. Okay. As peptidoglycan fragments can disseminate into circulation, so into the bloodstream, and prime the systemic immune response, so this is just the immune response throughout the whole body, the presence of these hydrolases, so SagA, and NOD2 active neuropeptides may be clinically relevant for predicting individual therapeutic responses. Okay, a lot more blah, blah, blah. Lastly, our findings corroborate a wealth of emerging evidence that NOD2 stimulation by MDP, the synthetic version of GMDP, so this is actually what companies are trying to make, this MDP, because they know, okay, it stimulates NOD2, which then results in a better immune response. So and basically any analogs. So analogs are just similar kinds of MDP-like peptides. So there's more and more evidence coming out from the scientific field that these MDP peptides can alter host immunity through multiple pathways, including direct activation of macrophages for tumor cell clearance. So some people think that macrophages can directly kill tumor cells. That's a little dubious, but it could happen. They also think that maybe when NOD2 is activated, there's this epigenetic reprogramming of monocytes. What does that mean? Basically, the epigenome, you could think of it as whether or not DNA is accessible or inaccessible. And there's basically, if signaling occurs into the cell, it can sometimes result in the DNA becoming coiled or uncoiled. And then if the DNA is coiled or uncoiled, this determines whether or not the gene can be made or basically be transcribed or oh my god. Anyways, and then they're like, oh, maybe these peptides can also generate type 1 dendritic cells and priming of dendritic cells for cross-presentation to CD8 T cells. Basically, dendritic cells are these special immune cells that activate T cells. So they think maybe somehow NOD signaling that's activated by the MDP peptides from bacteria is maybe somehow making dendritic cells better at activating T cells. So these little numbers are just referring to papers that they've linked in their references.
Why,
so let's
hello,
just...
my little coffee bean. Welcome
Okay,
to the
Tom.
Caffeine Club.
I wanted to say that in an Australian accent for some reason. Thank you so much for that sub. I appreciate you. Welcome into the Caffeine Club. So, yeah. Basically, the big takeaway is that there's these... So let's go back and do our little recap, okay? All right. So we learned that enterococci, which are these little bacteria that are found, they're usually found in our guts. We'll call them enterococcus. I don't know if they have a flagella or not. But basically, the enterococcus that's found, it's been found in patients who clear their tumors better. And then this is important, when they're given checkpoint blockade therapy. And I've already explained checkpoint blockade therapy earlier in this video. Basically, just think of it as an antibody that makes checkpoint blockade. Just think of it as an antibody that makes T cells better cancer killers. So enterococcus is more enriched in patients who clear tumors better when given checkpoint blockade therapy. This led them to ask, what is it about enterococcus that make them what is the enterococcus producing? And we found that enterococcus, in its little cute bacteria genome, it makes this thing called SagA. SagA is a hydrolase. So as a protein, it's like this little Pac-Man hydrolase. It cleaves the hydrogen bonds that hold together peptidoglycan. And peptidoglycan is what makes the cell wall on enterococci, but also other bacteria. So it cleaves the peptidoglycan, which is just like this sugar carbohydrate matrix. So here's SagA doing its thing, cleaving things, saggy cleavage, if that helps you remember it, great. And it cleaves it, and it makes these tiny little pieces of the peptidoglycan called neuropeptides, or oh my god, is it? So it's either GMDP I think that's what they were calling it. I'm already forgetting my acronyms. But it makes these little peptides called GMDP, which then activate the NOD2 receptor. So here, this is the NOD2 receptor on immune cells. These are not T cells. They're another, I don't know, they could be monocytes. This is just an immune cell. So then it activates it. And then this results in NF-kappa B activation. So the gene or basically the protein NF-kappa B, a part of it cleaves, and NF-kappa B is a transcription factor. So it turns on genes that are inflammatory. And we know that inflammation can help promote a good immune response in some cases. So it turns on inflammatory genes. And then somehow in an unknown mechanism that they haven't figured out yet, which might be paper number 2, they find that this results in more T cells that are cancer specific. And so they also see that the tumor growth curves, instead of the tumor growing out like that and escaping, this is a tumor growth. They see that with the neuropeptides that are produced by enterococci SagA, that the tumors are actually controlled. Tumor growth is controlled because the T cells and other immune cells potentially are killing the cancer cells better. So that is the summary of this paper, okay? So if you guys are just coming in here at the end and you're like, oh my God, what's this paper about? That's the summary. So yeah, anyways, wow. Whew. Let's see. I still haven't finished my 3rd cup of coffee.